Colleagues, good morning. My name is Aziz Sheikh. I'm the Director of the Usher Institute at the University of Edinburgh, and it's a real pleasure to welcome you to the 11th in our series of COVID-19 webinars. So just right at the outset, I'd like to thank my colleagues, Professor Linda Bald, and colleagues in the communications team, Jess Conway and Susan Buckingham, who've been instrumental in putting on this series. So as per previous sessions, this is available through Zoom and it's being live cast through YouTube. And so it will be available in due course. So please feel free to share details of this with colleagues. Because of the numbers of people who are joining, we won't be able to see or hear you directly, but we are very keen on your participation. So do enter your questions into the Q&A function. And then if colleagues could please upvote the questions that they're most interested in. What I'll try and do is ensure that those questions are covered over the course of the next hour. So I'd now like to introduce our speaker for today, and it's a real pleasure to welcome Professor Calum Semple. So Calum is Professor in Child Health and Outbreak Medicine at the University of Liverpool. He's also a member of the Scientific Advisory Group for Emergencies, or SAGE as it's known for those of you outside of the UK. So really providing pivotal advice to the government on the management of COVID-19 outbreak. He's together with Peter Openshaw and Kenny Bailey from the University of Edinburgh, is the PI on the ISARIC 4C COVID-19 work that's going on. And I'm really pleased that colleagues in the Asher, Ewan Harrison and Anne-Marie Doherty are playing a pivotal role in this. It's worth checking out Calum's Twitter feed. It goes by the name of @TweedyChap. And he's interested in bagpiping. I don't think we're going to get a rendition today, but maybe at some point in the future, Calum. He's a Scot, so a very warm welcome back, a virtual welcome to Edinburgh, Calum. Delighted to have you here. Thanks so much for making time. We know how busy you are and really looking forward to your presentation. So thanks so much.
Well, good morning, everybody, and thank you so much for inviting me to speak to you today. I'm going to tell you about a collaboration, the success of which would not have been possible without the collaboration and hard work of Kenneth Bailey, Anne-Marie Doherty, and Ewan Harrison, all members of the University of Edinburgh. It'll take me just a moment to screen share, and then we'll get into the talk. So here we go. Thank you very much. So I'm just going to... Thank you. I hope you can now all see my screen. According to my screen, it's gone green, so it's actually sharing the wrong one. So just give me a moment, I'll come out and start again.
So we can see your slides, Calum.
Thank you very much, Aziz. Thank you very much. So I'm going to talk to you today about ISARIC, which is the International Severe Acute Respiratory and Emerging Infection Consortium, and how we've been working to combat COVID-19. I've only put up a small number of the ISARIC 4C investigators. Our investigator list now numbers over 100 collaborators and investigators who are within our consortium. More information can be found at our website, which is isaric4c.net, and the study has got its own Twitter handle too. Pandemics are known to have occurred roughly every 23 years. We have, up till now, assumed that most of these have been influenza, but on closer examination and looking at the molecular clock of some of the viruses that we know about, there is suspicion that this outbreak here, possibly this one to the left, in the early 19th century, may have actually been due to coronaviruses. Pandemics are therefore certain, but we do not really know what the pathogen will be, what the disease will be, and how that disease will impact upon certain groups, society as a whole, the pressure on healthcare facilities, and the case fatality rate. Likewise, we won't know how diagnostics will perform, and if infection control will work. We don't know if we'll have current vaccines or therapeutics. And each of these questions has been challenged in the current COVID outbreak This slide was actually written several years before COVID, and I would have challenged you then to have said that we're going to have a virus that will spare children, will have a case fatality rate which is negligible really in children and yet so severe in the elderly, where our infection prevention control measures have failed so spectacularly, and where the disease process is really quite unusual, with both enteric, musculoskeletal and mucocutaneous features and distinct respiratory features as well. This is essentially to say that there is a strong justification for maintaining rapid research capacity. This slide illustrates the situation that we were in 2009, when the influenza outbreak and pandemic occurred. At that time, we got quite good observational public health research. In fact, it was public health physicians in Scotland that were most astute and identified the outbreak occurring in Dunoon and then in the southern part of Scotland. But despite that, preclinical research was slow and really only got going with a study called Mosaic in the tail of the first wave. The good observational public health research did start. That was a study called Flucin, and Flucin is the father of Cocin, which is a very successful study that I'll talk about later. But the preclinical research, which was run by Mosaic, only got going at the end of the study, end of the outbreak, and clinical trials just didn't happen. In fact, looking at the trials register, there was only one prospective randomized controlled trial conducted in China in 2009. The ideal situation is one where we have observational public health research right from the get-go, then with preclinical research following shortly afterwards and trials getting up and going. And for the first time in the last 20 years that I've been working on this, I'm going to be able to say that we have achieved this, and I think it's an amazing achievement. So how do we go about doing this? ISARIC is a small organization of infectious disease doctors, intensivists, both pediatrics and adults, who were frustrated by our inability to mount a coordinated research response and coordinated public health research response in 2009. Much in the same way as students of English literature will tell you, there's only really 7 stories in English literature. We looked at the landscape of clinical trials and studies and came to the conclusion that there were really only 4 different types of study that could be conducted. The first was a clinical characterization protocol. That would ask the who, what, and why. So who is infected, what does the disease look like, and the why, a little bit about pathogenesis. So if you like, that's the first of the stories that you can tell. The next is the standard drug trial, randomized controlled trial perhaps, or perhaps an open trial, perhaps an adaptive trial. That's a therapeutic trial. Third would be your vaccine study. The vaccine studies are very much harder to blind, require very large populations. To really prove they work, they need to be done during the outbreak to actually show you can prevent disease. It's a very different story to the drug trial and a very different story to the clinical characterization protocol. And lastly, the convalescent plasma trial. The challenges of giving convalescent plasma mean that really it's very difficult to run it alongside the other studies. So these are the 4 large trials and studies that you can imagine. And what the ISARIC group did was developed protocols for each of these studies. And we developed them to a high degree. So we had all the data elements identified, case report forms identified, common outcome measures agreed by consensus process. And we then took these to the World Health Organization for their ethics and regulatory approvals, cognizant that that doesn't actually count for much at a country level, but that on a global level, it's a good starting position from which to engage. Several countries then approved various combinations of these studies. And in the United Kingdom, the clinical characterization protocol was adopted by the urgent public health research process in 2012 on the back of recognition of MERS as an emerging threat. In these protocols and documents, we deliberately did not name the pathogen, and that was very important. This meant that in advance of an outbreak, all we had to do was ask for a quick amendment to any protocol for that pathogen to be listed. And we also put a clause in saying that these protocols were valid for, inverted commas, any pathogen of public health interest, close inverted commas, as decided by Public Health England or other public health authority. And this meant that as soon as the public health authorities in various countries acknowledged that they had an outbreak of public health interest, then this protocol could be activated without further recourse for ethical approval. Each of the protocols is tiered, and by that it's a layer cake. The lowest layer of each protocol was to collect data. The second layer was to do a single or limited biological sampling. The next layer up was to do serial biological sampling. And the top layer was to do intense high frequency sampling, perhaps to do population pharmacokinetics, or to look perhaps at immune modulators. The concept was to build this tier so that if availability of local resources increased, you could go up the tier. And if availability of local resources was constricted, perhaps due to other studies coming on board or staff sickness, people could go down to lower tiers in the protocol. And indeed, this is exactly what has happened in this current outbreak, where at the start we had 100% recruitment to tier 2 for the first 100 or so patients. It was quite remarkable. And that was done mainly in hospitals in the north of England, I have to say, and Newcastle particularly strong. And then as the outbreak got going and staff sickness occurred, we found sites having to drop down from tier 2 down to tier 1. ISARIC is now a network of networks, or a consortium of networks. We believe that we're covered in 130 countries, and we've got 43 networks that we work with. The global consortia is funded by a variety of different groups, and Bill and Melinda Gates have been particularly generous, as has Wellcome Trust, UK Aid, Zika Alliance, GoON, and GLOPID-R. In the United Kingdom, we're funded directly by NIHR and Medical Research Council. Oxford is the global coordinating centre. Here we see the list of our urgent public health research portfolio that was stood up in 2014. And this study at that time was called the Severe Acute Respiratory Infection Biological Sampling Study, which is a bit of a mouthful. But we had been just asked, this is how bizarre life is, we previously called it the Novel Coronavirus Characterization Protocol. But this has been criticized because it's been felt that a coronavirus was really quite unlikely to be causing a future pandemic, and we should really be thinking about flu, perhaps, how wrong we were. So, SARS coronavirus. Coronavirus, relating to the term crown, the electron microscopy of the virus shows that the virus particle is indeed encircled by these glycoprotein spikes. It's a tri-spike conformation, and it does give the picture of a crown, although I think it looks a bit more like a tiara than a crown. This is the map showing the evolution of SARS coronavirus as an emerging infection against the outbreaks of other coronaviruses. And this is why there's a suspicion that one of the outbreaks in the early 19th century was one of these coronaviruses too. Some of these coronaviruses, perhaps this one particularly, appears to have emerged from a bovine coronavirus. And of course, humans and cows have been closely working together. And there's a very good chance that this has jumped over to humans from the cows around about this time. So at least one of these outbreaks that we looked at in the previous pandemic is probably related to emergence of either 229 or OC43 in the early 19th century. This is where SARS-2 fits in 2019. Remember, we did have SARS-1, very closely related, in 2002, 2004, and MERS emerging in 2012. This is the timeline of the outbreak globally, so just at the tail end of 2019, we had the first cases of pneumonia of unknown etiology. Members of ISARIC, several of whom advised the government on the new and emerging respiratory virus advisory group. There's 3 ISARIC members on the UK NERV tag. That's Peter Horby, myself, and Peter Openshaw. And we're in pretty constant communication, and we were really getting quite anxious about this virus, particularly when we had cases outside of China. And we noticed the cases in Thailand, and then there was some in Singapore. And it literally took one email from me to the Deputy Chief Medical Officer on the 17th for us to get the go-ahead to stand up the study and activate the study, which, if you consider the amount of time it would have taken a few years ago to activate a national study, the amount of committees you'd have to go through. Now in 2020, a single WhatsApp followed up by a formal email, and we had a study open at 200 sites within 48 hours. That meant that when the first 2 cases were confirmed in the UK on the 31st of January, just 2 weeks later, they were then recruited to the study. And then from the end of January 31st through to the end of February, we had roughly 40 cases. So 40 cases in that first month. And then in the next month, we had 4,000 cases. So there you are, 1 case, 40 cases, 4,000 cases. And that rate of recruitment, I think, surprised us all. This is how the ISARIC study is working now. We have got 2 hubs, one in Glasgow, one in Edinburgh. Sorry, one in Glasgow, one in Liverpool. And these centres bring in the samples from our patients. We've now recently got a patient actually recruited from Inverness. And so samples are coming in from Inverness, as far as down as Bristol. They come to the Outbreak laboratories in Glasgow and Liverpool, and then material is aliquoted and sent to collaborators throughout the country for them to do the clever tests that they know all about. Edinburgh's particular role with Ewan Harrison and Anne-Marie Docherty and Kenny Bailey is the collation and the collation of the data and the processing of that data on an analysis platform, which allows us to link both the biological information generated by the research colleagues with the clinical information brought in from the hospitals. And that goes into a data safe haven. I should apologize, we've got 40,000 cases here. That's no longer correct. It's now 53,000 cases. And this is the kind of detail that we can generate. If you like, Public Health England takes the case epidemiology to the hospital door, and our study follows the patients then through the admission journey. We have over 2,500 medical students and research nurses collating data by hand from patients' notes, whether they be paper notes or electronic healthcare records. And this data goes onto a paper case report form and then is uploaded in the old-fashioned manner to a REDCap database. And then the analysis team in Edinburgh runs some clever scripts to clean it up and generate a most amazing report, which is provided online in a dynamic manner for various people to look at. And I'll talk about that a bit more in detail shortly. But this is our distribution of cases. You can see clusters where you'd expect in the south, in the London, Midlands. Then you've got the Liverpool, Manchester, Leeds conurbation, the northeast conurbation, and then the Scottish mid-belt. You can see recruitment accrual over time. Recruitment has now slowed down as we are past the peak of the outbreak. The red line represents men, the blue line represents women, and there's roughly a 60-40 split between men and women, and that has been seen in every other study that's been published. As I said, it's a deep data dig. The who is the demographics, the comorbidities, the presenting syndromes. The what is the treatments and the outcomes, and the why is the underpinning sampling, which supports other studies, such as the recovery study. Our dynamic report is a real-time analysis of this raw data. There's minimal cleaning. We try to get rid of the erroneous entries where somebody types in a patient's name as 1,090 years old, when really they meant the patient was 109 years old. We have had that happen. Initially, we used to say that the report gave us signals rather than messages, but now that the data set is so rich, we're now emphasizing the messages. Originally, the report was updated every 30 minutes, but with the size of the data set, that went to 4 hours, and now it's typically twice daily. The online report is accessed directly by the chief medical officers of the 4 nations and their deputies. It's accessed by the members of the Scientific Advisory Group for Emergencies. They all have login and they get it presented formally twice weekly. Scientific Pandemic Influenza Modelers Group, known as SPI-M, have API token access, and they're drawing the data out and doing some quite remarkable work around nosocomial infections, for example, and that has been very useful. They've also used the data to understand use of oxygen and intensive care beds. And our own public health agencies, that's the Public Health England and Public Health Scotland, have direct access to that data. I'm now going to give you a little bit about the results so far, and these results have been published in the British Medical Journal online just last week. We have been able to update the data just for you. This is a plot showing the sex split by age, and the colors give you the discharge, ongoing care, and those that have died. And it does illustrate well how men are far more badly affected than women, and that this effect occurs throughout all ages until you get to the age of 90. And that is only because women now outnumber men in surviving beyond the age of 90. So this phenomena is to do with female survival rather than anything else. We can look at the admission features in great detail. So we can look at the features such as cough, fever, and shortness of breath, which are the most predominant features. We did not include loss of smell or loss of taste because the case report form was developed well in advance of COVID. And we've only added in anosmia and ageusia into the case report as of 2 weeks ago. So we're not currently reporting that as a feature. But the main features are cough, fever, and because this is a hospital cohort, shortness of breath, it's not surprising to have that. We can look at the comorbidities in great detail, and we see that cardiac disease, obviously very high featured. Again, we did not capture hypertension as a separate entity because we did not predict that hypertension was going to be such a strong feature. And this is the problem of trying to predict the future. We don't always get it right. Interestingly, asthma, and this is chronic pulmonary disease excluding asthma, but asthma does not appear to be a strong feature of poor outcome or one of the strongest features of comorbidities. In fact, it's the non-asthmatic pulmonary disease that is stronger and cardiac disease. We have these scaled Venn diagrams or Euler diagrams, which show the overlap between the 3 most common symptoms. And you can see that Public Health England's original decision to use cough, fever, and shortness of breath was pretty good. If you just use cough, fever, and shortness of breath, you do pick up around about 95% of the cases that are now in our cohort. Part of the reason for not adding ageusia and anosmia at the earlier stages were that it would have affected our ability to have a standardized case definition throughout several epidemiological studies. And adding it in partway through the outbreak was not particularly helpful for people doing epidemiological studies. On the right side of the screen, we see the 3 most common comorbidities. And it's interesting that there's far less overlap than I thought there would be. In fact, they do almost behave like 3 quite distinct risks. I would have assumed that there'd be far more overlap, for example, between diabetes and chronic cardiac disease, but it's just not the case. I particularly enjoyed this kind of analysis, which looks at the clusters of symptoms associated with an emerging disease. What we find here is that there is in general terms, a box up here, which is associated with more traditional respiratory problems with particular grouping around cough, shortness of breath, and fever. But there is a distinct cluster of joint pain, myalgia, fatigue, and headache, and another discrete cluster of diarrhea, vomiting, and abdominal pain, and then another group of mucocutaneous and perhaps neuromucocutaneous features down here. When we look at the clusters in the round about a quarter of the people presenting with classic COVID, that's the respiratory complications, round about a quarter of the people with respiratory COVID have some degree of enteric feature as well. And round about 5% of people have an enteric feature only, which means that people are being admitted to hospital with acute abdomens, diarrhea, and vomiting, and it's not immediately obvious that they have COVID disease. And until quite recently, these people were misclassified and placed on non-COVID wards because they didn't meet a case definition, and consequently they were the sources of in-hospital acquired COVID infection. The granular detail that's now possible in the study is remarkable. We can do essentially a chop on the time series of data so that we simply look at a couple of weeks in isolation, and so that we can see how healthcare resources have changed during the course of the outbreak. And so this is the cut that was taken in the time period more than 14 days ago and less than 28 days ago, to look at the types of care that are currently being used. This column shows the first place of admission. So you can see that relatively few people are now currently going straight to intensive care, the majority of people going to the ward. But after that day, people are still deteriorating, and the numbers in critical care have roughly doubled. We can then see what is happening to these people who are getting high flow oxygen, those that are on non-invasive ventilation, and those that are on invasive ventilation, and then follow them to outcome. If we look at the same type of analysis for the whole period, there's far greater use of oxygen. And this is because we've moved away from the majority of people having acute illness to a situation where many of the people in our study are actually recovering and convalescing in hospital. This slide illustrates that point. We show the outcomes for all admissions who've been admitted more than 14 days ago. This group here are those people that have died, this group those that are in ongoing care, and this group those that have been discharged alive. And this is a rather sobering figure, because at present we would say that a quarter of our patients have died, and around about a third are receiving ongoing care. There's a slightly different way of looking at the data where we look at patients for whom outcomes are assumed, and this figure rises up to around about a third. This figure then drops down quite a bit. The outcomes for those people requiring invasive mechanical ventilation are also quite sobering. And you can see that for those who we've got very firm outcomes, 40% have died, and only a third have been discharged alive. Survival is clearly defined most strongly by age, with those people under the age of 50 having a very low risk of death. But sadly for myself, being 52, slightly overweight and with hypertension, I'm definitely in this high risk group. And I wouldn't have thought that that was such a strong feature, but in fact this virus is very nasty, highly infectious, and as you've seen from the last two slides, it's got quite a high morbidity for those in hospital. So don't be fooled. Don't believe anyone that says this is just a bad dose of the flu. It's not. This is the multivariable Cox proportional hazard model developed by Ewan Harrison and Anne-Marie Doherty at the Usher Institute, and which is probably the top most interesting figure in the BMJ and the one that's getting most attention at the moment. it shows very nicely the hazard increasing with age, and how for each increase in age, the hazard actually increases quite a lot more. So with each 10-year group, the hazard goes up. We also show that women are about 20% less likely than the standard risk to have severe disease. Cardiac disease, pulmonary disease, kidney disease, and malignancy are perhaps obvious comorbidities where you might expect these to increase your risk of death. But obesity, even when taking into account the other features, obesity is a very strong risk factor. Again, around about a 20% increased risk of hazard against standard. And this is a feature that was not well described in the data that came out of China or other studies. And this has led to questions around shielding policy. And indeed, obesity is included in the ultra-high risk group for those people with BMIs over 40. But we did not classify obesity by BMI. We classified obesity by physician-assigned obesity. And that could be probably anything between 30 and 40, indicating that the current shielding criteria may actually be a little bit conservative and probably should reflect that people that are in the BMI 30 through to 40 remain at higher risk. This is a slide which I barely feel competent to describe, as there's far greater expertise, particularly with our chairman, Professor Aziz Sheikh, who has been involved in this work with us and one of our collaborators. Essentially, we've been looking at the effect of ethnicity and outcome in COVID disease, and we've taken great care with this analysis, and it's perhaps a set of analyses that has been more closely scrutinized than anything else that we've done to date. This kind of analysis is actually perhaps not the best way of showing the data. A diagram that I'll show next is probably better, but it does show the relative effect size. So what we have done here is taken into account ethnicity, sex, deprivation, diabetes, and the other comorbidities, and attempted to adjust for all these risks. And even when we adjust for all known mediators, we still find that the population from the South Asian ethnicity are at significantly higher risk of poor outcome. And by significantly, I mean both statistically, but in a clinically meaningful manner. That's a 20% increased risk of hazard compared to the standard risk. And that takes into account age, sex, obesity, diabetes, and these other known confounders. Perhaps a better way of showing this diagram, I'm told, because this is certainly outside my area of expertise, are these diagrams which show the modification of age, sex, and race on particular disease comorbidities, because it's not a straightforward linear process. Ethnicity has some genetic features, as does deprivation upon race. And so you have quite a complicated situation, and this kind of diagram allows a better exploration of the risk factors. And indeed, ethnicity does appear to have an influence on risk of diabetes. But the story is quite complicated, because if we look at the risks about ethnicity, and this looks at ethnicity across ages, we find that the difference between diabetes between the white population and the South Asian population is most pronounced in the younger age groups. And yet the differences in age, essentially the risk of mortality is affected by diabetes in a younger age group, but there are significant differences in an older age group, which are still not explained by the presence of diabetes. And it's a bit of a, it gives me a bit of brain ache trying to work out what's going on here. But essentially, diabetes is quite different in the younger age groups, whereas age is more important in the older age groups, but it's still the difference between ethnicity is very obvious. So only some of this difference can be accounted for by diabetes, if you like. There's still much unknown. So to conclude, recruitment to urgent public health research is feasible now in response to novel pathogens and during a pandemic. Our ability to respond was mainly due to rehearsals and activation for cases of MERS and monkeypox that occurred in 2018. And we did a rehearsal also in 2016. We responded to the Ebola outbreak in 2014. And I think if we hadn't kept the study activated and rehearsed for these events, we wouldn't have been able to step up so quickly in the outbreak of COVID. So here's our diagram from the start, where we couldn't do trials. Here's where we are now. We did achieve this. The ISARIC study got up and going. And in fact, we were actually going right from the start. So I can move this one back a little bit because we were right there from the start. Preclinical research through the biological sampling for the ISARIC study, also right from the start. And Peter Horby's recovery study, which is also an ISARIC study, has now recruited, I think, 10,000 patients in England and Wales and Scotland. And consequently, we have now got a situation where clinical trials can be initiated in time to make a difference during outbreaks. The number of people I need to thank is just so large. As I said, we've got 2,500 research nurses and medical students. We've got over 200 local investigators at sites, over 166, I think that might even be closer to 200 now, R&D administrators. We've got legal teams in Liverpool and Edinburgh. We've got finance teams in Liverpool and Edinburgh. We've got multiple educational institutions also supporting us. A lot of what's been done has been done through the Office of the Chief Medical Officer, just telling people to get on with it, which is very useful. If you'd like to follow us, CCP UK, that's Clinical Characterization Protocol UK Study, and that's our website. And that's my 40 minutes, and thank you ever so much for this opportunity to talk about my favourite topic.
Great, thanks so much, Calum. Absolutely fantastic presentation, and clearly an outstanding resource that you and colleagues have created. So thank you so much really for this. So Calum, there are a number of questions that have come in. I'll just work through these. So the first is, do you have any insights as to when the first case was in Scotland? And specifically, the questioner is asking, was it Dundee or was it linked to the night conference in Edinburgh? Any insights from ISARIC or otherwise?
Okay, so I'm going to have to plead ignorance, but I would also say, I truly am ignorant about this, by the way, but I would also say we do have to be incredibly careful as outbreak investigators to not identify individuals by inference. So normally I wouldn't have talked at all about the cases in Newcastle, but the cases in Newcastle were highly publicized in the media, and consequently I could never be accused as a GMC registered doctor of breaking confidence around the issues around the fact that these cases occurred. And recruitment to our study does not require consent. It's done on the basis of depersonalized healthcare records. So we currently have 40% of all of the cases in the United Kingdom in our study. So nothing I've said can actually identify people or break ethics. But if I was to start to say, oh, the first case in Scotland was at a conference on this day, then that's something that's not in the public domain and would lead to embarrassment and also erode trust in the kind of research that we do. So it's a question that I genuinely don't know the answer to, and even if I did know the answer, I wouldn't tell you.
Great. Okay, thanks, Calum. So there's another question around pregnancy-related outcomes in the case report form, and a related one around whether you've got data in the ISARIC data set on children.
Well, so first of all, yes, we do. And we've published a little bit about pregnancy in the BMJ paper. Round about 10% of the women in the cohort were pregnant. And the proportion expected in the population is around about 8% by prevalence of pregnancy in the possible pregnant age group, which it can be worked out. So we're not seeing an excess of pregnant women as a risk group. We're not looking particularly at pregnancy outcomes. That is a separate study run by UCOS. But as far as we can see, pregnancy does not appear to be a high risk group for whom we would be recommending for particular shielding. I know that's contrary to guidance, but that's the nature of guidance. Guidance has to be developed quickly at the start of an outbreak, and they didn't benefit from the knowledge, the scientific knowledge that we and others have generated. As far as children go, yes, we've got, at the last count, we had around about 400 children in the data set, and we had biological sampling from 47 of them. And that's round about 90% of all the children that have been admitted to hospital. So in fact, whereas 40% of the adults admitted are in our set, we've got close to 90% of the children, and they've had very good outcomes.
Great, fantastic. So there's another couple of questions around risk factors. So the first is whether smoking is an independent risk factor for poor outcomes. And the second is around obesity, and specifically the question is the relationship with obesity, as your understanding, is this causal or not?
So the first question about smoking, I think this was following an article in the Telegraph, and I was asked about this yesterday. We've not looked at smoking in detail. I'm sure it's something that we'll be scurrying away to do now, and I'm sure Anne-Marie and her colleagues will be putting a master's student onto it straight away. I can't comment, we've not looked at it. As far as obesity, what do you really want to know about obesity? It's none of these, because obesity is a sort of a scalar issue. It's a sort of, you can be slightly overweight, you can be obese, you can be morbidly obese. I think what we need to take away from the data is that a clean cut at 40 is not a cliff edge. If you're slightly overweight, that's not good. If you're very overweight, that's really bad. The mechanisms, we don't know if it's to do with weight or fat on the chest and the abdomen inhibiting respiration in extremis, or whether it's to do with the inflammatory environment that adipocytes encourage. But it's a real effect, and it appears independent of our other known modifiers and confounders.
Great. So there's a question, Calum, around potentially additional risk factors identified subsequently that weren't captured in the CRF. So, for example, blood type. Would there be any possibility of retrospectively collecting this information and then looking at those risk factors?
So we have been collecting blood type, and we haven't done an analysis on it. There are many more data points in the case report form, and people can download it and have a look at it and suggest analysis or apply to have access to the data set to do their own analysis. We welcome that. It's difficult to retrospectively add stuff to the case report form, but I'm sure your institute knows better than anyone that linkage to other data sets is possible. And we have now requested for CHI numbers and NHS numbers to be added to the existing data. And that's been done at direction of the chief medical officers for all 4 countries.
Yeah, I mean, I think that will open up all sorts of additional possibilities. So I mean, I think it's a really smart move. So thank you for doing that. So there's a question from Lawrence Greer, who's in the Usher, and his question is, have you noticed any change in clinical practice for those with the most severe disease? For example, is there a change in the kind of threshold for mechanical ventilation?
We did see changes in patterns of resource use over the course of the outbreak, and others are better qualified to describe why that might be. But it is reasonable to assume that there were improvements in advanced care planning that led to more appropriate decisions about who went on to receive intensive care. Anne-Marie Doherty has explained to me that people are not sent to intensive care units to die. They're sent to intensive care units to recover. And if their disease is so severe that a period of intensive care is highly likely to be futile, then that is unlikely to be in their interests. And so that has changed. But we are aware of reports of changes in management in intensive care and general wards, increased use of non-invasive ventilation, increased use of proning of patients as well. So there's a combination there of better care and better planning of care, of that care.
Great. So there's a linked question from Tim Walsh. And really, as we move into this kind of chronic phase of the illness, and chronic is put in inverted commas, do you think there will be a change in thresholds or criteria for people being admitted to critical care settings? Because, I mean, this has obviously been quite a contentious issue so far.
I really can't answer that question. I'm a pediatric respiratory consultant. I'm doing observational research. I think, as Tim knows, that's quite a political question, and one that's probably better answered by adult intensivists. Yeah, so Tim, maybe connect up with Anne-Marie on that, but a really important question. So the next one is from John Frank, and John asks around the issue of deprivation and what signals are you seeing in that respect? Is deprivation emerging as an independent risk factor for poor outcomes? I'm not going to answer that directly. I will say that COVID has revealed deprivation and made all inequalities worse. And deprivation is a risk factor for severe outcome. We have included it in our models, particularly our models around ethnicity. So I think the straight up answer is yes, it is. But when you're looking at environmental insults and socioeconomic insults, it has to be taken in the round along with the other issues that are going on in the community. And that's where we benefit from collaborating with people like Aziz and Ian Sinha, who've done much more work in this area than I have. Great. So a few final ones, Calum, but thanks for kind of working through these. There's a question from Richmond Davies around the ethnicity work, and what Richmond is asking is whether the signal is the same across the UK nations, and specifically, what are we seeing in Scotland in relation to ethnic variations in outcomes? Well, really, you should be answering that, shouldn't you, Aziz? Because you've just put in a really nice grant working with some excellent collaborators from south of the border on this one. So what do you say to that one? Yeah, so I mean, at the moment, Richmond, the analysis that Calum presented was for the 3 UK nations, England, Wales and Scotland. I mean, I think the trouble that we run into in Scotland is small numbers to see any reliable signal, but there will be data emerging in due course, so do watch this space. Right, I just want to try and make the most of having sort of Calum's expertise with us in the last few minutes. So there's a question, Calum, around long-term outcomes, and it's really, do the public understand well enough, in your view, the kind of long-term implications of COVID-19 in survivors? And you may be able to say something around the bid that's just gone in with Chris Breitling and colleagues. I don't think the public have a clue about the long-term consequences of COVID. I think people struggle to even understand that an episode of pneumonia will set someone back 6 months and take a long time to recover. This is a disabling disease. It is generating a population of people that are very damaged and are going to require considerable rehabilitation. And that message has not gone out there yet. Chris Breitling has a grant going in to look at respiratory follow-up, and that has an element of follow-up on disability. Our team in Liverpool, working with Edinburgh and Imperial College, have got a grant going in to look particularly at fatigue and the possibility of chronic fatigue syndrome. You'll remember that slide I showed, which shows a clustering of symptoms, which are myalgia, fatigue, and then the neurological grouping, the neuromucutaneous syndrome. And that would, to me, be a warning sign that there likely is going to be a chronic fatigue type illness, because we are seeing direct neurological and mucocutaneous features. And that the neuromucocutaneous features, which you see with other diseases like Kawasaki-like illnesses, but also we saw it with Ebola syndrome. That was strongly associated with post-Ebola syndrome, which included muscle ache and fatigue and quite significant neuropsychiatric and psychological problems. So I think we have got a new disease, and we'll have a post-COVID syndrome, a collection of disorders that we will be seeing.
Great. Calum, there's then a question from Richard Simpson, and he's asking, can you say a bit more about the international collaboration? So I think at the outset you mentioned 130 or so countries involved in ISARIC in some way or another. How many of them been activated in relation to COVID? What's the plans for sort of integrating data across these countries?
So the ISARIC Global Coordinating Centre in Oxford uses a REDCap database, and the same case report form that we use in the UK is used in the other countries. So that the largest study to come out of China, that was 40,000 cases, used the same case report form as we did. Not all countries are comfortable to share their data. And off the top of my head, I think we've got 14 countries currently sharing data, which has been pooled in Oxford, analysed by Crystal Donnelly. And the group in Edinburgh, that's Anne-Marie and Ewan and Crystal Donnelly's team, have been sharing code. And there is a joint analysis that gets sent on a weekly basis out to WHO and is posted on the ISARIC website. And that I think the UK now accounts for around about 95% of the data in that data set. But all the other countries are finding very similar data to ours. There's been no shockers, no surprises. No one is saying, look, this is a disease of children or women. Everybody's saying this is a disease mainly of elderly and mostly affecting men. So that the story is consistent.
Great, okay. And so Calum, the last question, and this is coming from two colleagues, so Alan Archibald and Yiming Hu from, Yiming's joining us from Malaysia. So absolutely fantastic data set, direct connectivity with government agencies, chief medical officers. And so really the question they're asking is, what impact has this had on changing the shape of the pandemic in the UK?
I'm not sure that it's changed the shape of the curve. I think that is down to the population's adherence to advice on social distancing and staying at home. And adherence to that advice has been critically important and wholly dependent on a social contract between the government, society at large, and our health service. And maintaining that social contract is incredibly important. Where I think our data has made a difference is we were the first signal of a hospital-acquired infection problem, and that led to closer scrutiny of infection prevention control procedures. We were able to show healthcare resource use, the provision of oxygen particularly, and intensive care beds at a very early stage of the outbreak, and the issue around about obesity and other risk factors, which has led to closer scrutiny of the shielding guidelines. Going forward, I'm hoping that our data will inform the vaccine strategies, because we're clearly going to be able to identify the real risk groups rather than the imagined risk groups that are based on past experience with influenza.
Great. So Calum, just an enormous thanks for joining us. And not only that, for everything that you're doing. I mean, it's absolutely fantastic work. And it really is a privilege on behalf of colleagues in the Usher to be collaborating with you and your team. We look forward to welcoming you in person in due course back to Scotland. But Calum, thanks so much again. And what we'll do is we'll share these slides with the Scottish Government as we're doing which presentations.
Thank you. And could I just extend my thanks?
Great. Thanks so much, Calum. Thank you.
Thank you.
Thank you. Thanks, Calum. And Jess, if you could bring up the slide, please, of the next webinar that we've got. Good. So colleagues, thanks so much for joining. I hope you're finding this useful. It was an absolutely fantastic presentation from a real leader in this respect, and we've got 2 further webinars lined up for you. So the next one is from New Zealand. We'll be hearing from Michael Baker on Thursday, the 11th of June. And then following that, we'll be hearing from Naveed Satara, a close colleague from Glasgow, who'll be doing a deep dive into obesity. So clearly one of the questions that come up. So we're going to call it a day there, but thanks again for joining. Take care. Look forward to meeting you again soon. Thanks so much. Goodbye.